All right. Hello, everyone. My presentation is on mechanisms of IgG. I prefer to say this than immunoglobulin, just because I struggle to pronounce it. IgG-mediated opsonophagocytosis of extracellular bacteria. So for some major learning points, extracellular bacteria are a major cause of infection, and antibodies help eliminate these pathogens through a process called opsonophagocytosis, which is just a combination of opsonization and phagocytosis. This presentation will help explain how these components, IgG antibodies, complement proteins, Fc receptors, CD38, and neonatal receptors, work together to promote bacterial clearance. So for a background, why do we need opsonophagocytosis? Many extracellular bacteria resist direct killing. Phagocytes cannot effectively recognize all bacteria on their own. So antibodies and complement act as tags that mark bacteria for destruction. This whole sentence right here is just the process of opsonization. Opsonization literally means to prepare for eating. Antibodies make pathogens easier for phagocytes to find and ingest, and this process is an important component of vaccine-induced protection. So this image I made in Canva um, shows just a regular bacteria without like anything special in it. And then this is a bacteria covered or coated in the IgG antibodies. These IgG antibodies help a phagocyte recognize it, which helps with the opsonophagocytosis process. So for an overview of the IgG-mediated opsonophagocytosis, this may look like a lot, but I'm going to go through it right here. So these figures summarize the entire process. We'll start with 1. IgG antibodies bind bacterial antigens and complement deposits C3b. So on this image right here, oh my gosh, it's overlapping. So we'll see that these antibodies right here are going to bind to this extracellular bacterium, and there's this C3b deposited on the surface. So these Fc gamma receptors and this CR2 receptor on this phagocyte are going to recognize this bacterium because it has these tags on it, this opsonization that's occurred because of these things that are on it, right? So the Fc gamma receptors and complement receptors recognize the opsonized bacteria, and CD38 signaling promotes engulfment via enhancing signals. So right here, we'll see that um, I drew this because Canva was giving me some issues, so this kind of looks crappy. But after the recognition happens, the engulfment process is going to be enhanced because of the CD38 here. So like this is without CD38. It's giving a weaker engulfment signal, but the signal is enhanced and stronger because the CD38 is here to encourage that, right? And then we'll see here that the neonatal FcRn receptor enhances internalization. So this is the next process. Like we'll see, this is like recognition, engulfment, and then internalization. So this is without the FcRn. It's just taking a little bite of it. This is with FcRn. It is taking a big bite out of it because it's enhancing that internalization process. It's pushing it towards the center. That's what internalization means to like push something towards the center, right? And then the sixth step doesn't really it's not a mechanism, it's just like the final step. That bacterium is destroyed in the phagolysosome, whatever, how to pronounce that. And this is just a simplified summary of the process. So this is a little bit redundant, but what is opsonization? Opsonization is the coating of a pathogen with molecules that promote recognition and phagocytosis by immune cells. This should be basically pretty similar to what we learned in class. These are the components that this process includes. So these are some things that we'll need to know in order to kind of look at the papers, the figures that I'm going to describe. And from lecture, we learned IgG binds specific bacterial antigens, complement activation deposit C3b, both act as opsonins, and phagocytes recognize these molecules through surface receptors. Think of this as like a flipped classroom type thing, like I'm teaching you these things first so you can understand the figures. So this is figure 1 by Rikita and others from the year 2000. The figure title is Specific Antibodies and Complement Promote Bacterial Binding. So they looked at this bacteria strain or bacteria called Enterococcus faecium, and it's resistant to phagocytosis. And they want to know, can antibodies and complement promote PMN binding? So for the methods, the bacteria were incubated with an immune serum. It's called like the immune rabbit serum, the IRS, or control serum, and exposed to PMNs. So PMNs are they're bacteria. It's like the attachment of a bacteria to leukocytes, which are like mostly neutrophils. And we'll start with panel A here. I'll describe it. You don't have to worry about the rest of this. So IRS absorbed with TX0016. That's a strain that is resistant to phagocytosis. And we see that the PMM binding drops dramatically. This is measuring the PMM. This means that the TX strain removed the antibodies responsible for opsonization. And then we look at this IRS RLA3 one. This is the susceptible to phagocytosis one. The PMM binding stayed really high, which means that the RLA3 did not remove the important antibodies. So something in the strain that is susceptible to phagocytosis is allowing it to bind to stuff. And if we look at panel 3, we are looking at 3 levels of IRS, the rabbit serum, where pure IRS is where the antibodies are free to bind to the TX strain of bacteria, which is resulting in high bacterial binding to PMMs. So this is a positive control. The next one is the IRS plus carbohydrate, and it's the purified carbohydrate absorbs many of the antibodies, and these antibodies can no longer bind to the bacterial surface. So this is resulting in the PMM binding dropping dramatically, suggesting that the most important opsonizing antibodies are recognizing a carbohydrate antigen on the TX strain. And then the IRS plus surface proteins one, the surface protein extract removes relatively few of the opsonizing antibodies, and most antibodies remain available to bind bacteria, which is resulting in the PMM binding remaining relatively high. And this suggests that the surface proteins are not the primary targets of the antibodies responsible for opsonization. So all of this breeds the conclusion that the opsonizing antibodies primarily recognize a bacterial carbohydrate antigen. So does this answer the question? Yes, it does. Specific antibodies and complement coat bacteria and promote PMM binding, basically. And now we're going to look at figure 2 by Kang and others from the year 2012. The figure title is CD38 signaling is required for efficient phagocytosis. And for the background, they looked at Fc gamma receptors triggering phagocytosis, but signaling pathways are unclear. So does CD38 contribute to uptake of the IgG opsonized bacteria? And for methods, they looked at wild type and CD38 deficient mice, and they received opsonized BCG. BCG is a live reduced pathogen commonly used in the tuberculosis vaccine. So panel A shows that significantly fewer macrophages. This is negative plus plus, right, by the way. Fewer macrophages from CD38 knockout mice contain BCG bacteria compared with the wild type macrophages. And panel B confirms this result in the wild type macrophages. This is plus plus minus minus. Sorry, I lost train of thought. Panel B confirms this because the anti-BCG staining shifted the fluorescence to the right, like this red one's a little bit to the right. It's not completely overlapping the blue one like this one, and the cons means control, so just keep that in mind. Indicating that bacterial uptake is happening here. In contrast, the CD38 deficient macrophages showed almost no shift, suggesting little or no phagocytosed bacteria. Together, these results demonstrate that CD38 is an important signaling molecule required for efficient Fc gamma receptor-mediated phagocytosis of the IgG opsonized bacteria. And for our last figure by Vitterson and others from 2006, the title is FcRn enhances internalization of IgG opsonized bacteria. And so they knew that FcRn was known for IgG transport, but does FcRn contribute to phagocytosis? Researchers compared the wild type of IgG and FcRn binding deficient mutant with H435A IgG. I'll just call it mutant because that's a mouthful. So panel A demonstrates that the mutant antibody still binds bacterial antigen normally. Like they overlap perfectly. There's nothing different there to the normal one and the mutant. This allows researchers to isolate the effect of ICRN. And panels B and C are kind of showing the same thing. They show that the bacteria coated with the mutant IgG binds normally to neutrophils, but are internalized less efficiently. Like this one looks kind of messed up and this one looks normal. Right here, we see that the mutant is showing lesser rates of phagocytosis in the ingested, but are showing like regular like binding things. So it doesn't really have anything to do with binding. It's mostly like in the ingestion process. Panel D confirms this finding in FcRn deficient mice. It shows that in the bound one, FcRn is not needed for bacterial attachment, and that in the ingested one, the loss of FcRn decreases bacterial uptake. So together, these results demonstrate that that FcRn is not required for bacterial recognition, but enhances the internalization of IgG opsonized extracellular bacteria, which makes phagocytosis more efficient. Like it's not needed. Like this is still kind of happening at a rate, but it's just not as high, right? So it's more efficient. So this is basically a review of what we just went over. We can connect this to the papers more directly. So from the, what's his name? Rikita. From the Rikita paper, we see that these little components, the antibodies and the 3C, C3b deposit on the surface of this bacteria are needed for the recognition on this phagocyte. This phagocyte uses its Fc gamma receptors and the CR1 receptors to identify and recognize this bacteria. And then from the Kang paper, we see that this CD38 is needed to enhance a stronger signal for the engulfment process. And then from the Vitterson paper, we know that FcRn, the neonatal receptor, is needed to make efficient engulfment, or I guess internalization. And so I'll briefly go over this because I think I'm running out of time. Clinical significance, why does this matter to us? We need it for vaccines to generate high affinity IgG antibodies, for therapeutics, for infection control. And to connect some of the stuff from lecture, we saw that long-lived plasma cells maintain protective antibody levels, and vaccine-induced IgG can provide long-term protection against extracellular pathogens. So, not to be too redundant, but here's some key takeaways to help you answer the questions I have for you on the discussion. IgG antibodies opsonize extracellular bacteria, complement C3b enhances bacterial recognition, Fc gamma receptors recognize opsonized bacteria, and CD38 signaling promotes phagocytic uptake. FcRn enhances internalization and bacterial destruction. And so, as a final summary, IgG-mediated opsonophagocytosis is a multi-step process requiring cooperation between antibodies, complement proteins, cell surface receptors, and intracellular signaling pathways to effectively eliminate extracellular bacterial pathogens. So my presentation is just on the mechanisms, all of these mechanisms, all of these components that help opsonophagocytosis occur, and their roles in the whole entire process. I hope this paper wasn't too redundant, and I hope I explained the papers well enough. Please let me know if I messed anything up, and thank you for listening.